The overall goal of the following experiment is to introduce oncogenes, or other potentially hazardous genes, into human cells using Ecotropic Lentivirus. This is achieved by producing pantropic lentivirus encoding the mouse retrovirus receptor, as well as ecotropic lentivirus encoding the gene of interest. Cells are transduced with the mouse retrovirus receptor, which makes them susceptible to transduction with ecotropic virus. Next, the cells are transduced with ecotropic virus in order to introduce the oncogene, using polymer-complexation with polybrine and chondroitin sulfate to enhance transduction. Results are obtained that show selective transduction of human cells with ecotropic lentivirus based on fluorescent microscopy and FACS.
The main advantage of this technique over existing methods, such as gamma-retroviral transduction, is that lentiviral particles can more effectively transduce many cell types, including non-dividing cells such as stem cells.
This method can help answer key questions in the field of cancer and stem cell research, such as understanding the function of potential oncogenes.
Before beginning this procedure, consult your institutional safety official and follow their recommended safety guidelines. Begin with a culture of healthy, rapidly growing 293 T cells. Working in a tissue culture hood under aseptic conditions, resuspend 5 × 10⁶ 293 T cells in 10 ml of antibiotic-free 293 T medium. Pipette the cell suspension into a 10 cm tissue culture plate and incubate the plate overnight in a tissue culture incubator at 37 degrees Celsius and 5% carbon dioxide. The next